Hello on this lovely snowy day. It is starting to snowfall again as I record this, so I hope you're staying warm and cozy. I wanted to just show you some stuff that we have on our Blackboard course for you for under the lab resources folder at the very top is this midterm lab practical study guide. I did not make this. Other students made this from previous sections, but it's still very relevant. It basically takes all the learning objectives that are at the beginning of each lab, and it kind of lists those out. And then the students went in and they filled them out. They added in details. They defined terms for those where there were actual experiments. If I scroll down to the pH and buffers experiment. It broke down the different exercises. It talked about the different controls, just as a way to hopefully help jog your memory to kind of summarize some of this stuff for you. There's the stuff for microscopes. This is a very helpful study tool. I still recommend make sure you're actually reading these labs, go back through them and read them. Make sure you understand what the experiments were trying to test, what were the reactants, what were the methods that we used, what was the science behind them. Look through the PowerPoints that I post onto the lab resources. Go back through any of your quizzes, your homeworks, those kind of things. Make sure you're very much understanding. You could explain these labs to somebody if I just said, okay, explain your diffusion osmosis lab to me. What did you do? What was the point of it? What's the science behind it? You should be able to talk me through those things. What were the tools that you used, right? Those kind of questions. For those of you that are missing lab today, under lab unit 8, here are your lab makeup activities. If you did not miss lab this week, if you are section 2240, you don't have to complete this. So just kind of being aware of that. It's not for a grade, it's for attendance. That's how I'll track it. If you are in section 2240 and you want to go through it, you can. It won't give you extra credit or anything, but it's there. Down at the bottom under the Unit 2, Chapters 5 and 6, I did add some things into the Chapter 5 learning resources, and some of these activities will be part of your makeup for today, but just go ahead and know that those are there and available for you. And this video will be found in this folder as well, which you're watching this video, so you probably have already found it. That's weird. Can't think about that. All right, let's get into the lecture. Okay, so today we are going to continue our lecture on photosynthesis. If you remember, we kind of finished up last time going through some of the basic overviews, talking about the structures of a leaf, the structure of a chloroplast, and just kind of introducing what is actually happening in photosynthesis. What is the overall reactions? What are we trying to be produced in this process? So if you need a jog on that, go ahead and pause this video, jump back over, maybe click into those chapter 5 resources, watch some of those videos, just kind of overview process of photosynthesis before you come into this one, because I'm going to kind of be jumping into some of the big science. We will do a review when we get back into class in person. We won't just drop this. We'll have a little bit more time to kind of talk about these processes. So as a reminder, photosynthesis is this series of chemical reactions. Your reactants are light. Oh, you know what? I'm going to need to adjust real quick. Okay, I think this will let me. Yeah, great. That lets me click. Okay, photosynthesis, right, is a series of chemical reactions. There are two different reactions. You have light reactions and your carbon reactions. So this graph we're going to come back to multiple times because it really does a good job of summarizing that. Within the chloroplast, you have these stacks of thylakoids, right? And thylakoids are where the process of photosynthesis happens. You have your light reactions where you have light and water being used. Your inputs are light and water, and your products, your outputs are oxygen, ATP, and NADPH, right? We said these are the high energy carriers. ATP, we can use that directly for energy, whereas the NADPH, these are carrying these electrons that we need to actually reduce our carbon. So over here we have our carbon dioxide that gets brought into the carbon reactions, and then we use the energy from ATP, we use the electrons from NADPH, and we can reduce it into sugar. We call this our carbon fixation. So we'll use that term a lot. You'll hear me say a lot, carbon fixation. And so literally we're fixing carbon dioxide into an organic molecule. We're taking inorganic carbon and turning it into sorry. We're taking inorganic carbon and turning it into organic carbon. That's a pretty impressive process. Well, plants are. We don't do it. Okay, so good summaries, right? The light reactions occur in the thylakoids, require water and light. Oh, how did that? It did not apply to all slides. I am so sorry. Okay, that should have fixed that. Great. So the light reactions, right? I would recommend writing a summary table. This is what we're going to do in class next week too, but go ahead and do it on your own as well. But the light reactions occur in the thylakoids. They require water and light. Your products are ATP and NADPH, and your oxygen, your O2 gas, is a byproduct. It's a waste product in this process. So how does that actually work? How do the light reactions work? And this picture is pretty busy, but we're going to break it down into three kind of main sections. But just to kind of see this big overview, what we have here, this membrane, these phospholipid membranes, this is your thylakoid. Okay, you have the space outside of the thylakoid, and then you have the space inside of the thylakoid. Okay, you have all of these photosystems embedded in it. So remember, we said that there are two photosystems in a chain, the photosystem 2 and the photosystem 1. They were named in terms of how they were discovered, not their order of operation. So photosystem 1 was discovered first, but it's actually the second photosystem in the process, whereas photosystem 2, PS2, that was discovered second, but it's actually the first one in the process. So it's just how it goes. So these comprise what we call this electron transport chain. And remember, photosynthesis is a series of redox reactions. We're moving electrons back and forth. And when we do that, we're oxidizing, reducing different molecules, and we're able to harness some of that electron's energy, release it and harness it for different purposes for work. So let's kind of go through, again, just kind of keeping in mind, okay, here's this chain of photosystems with these molecules in between. All right, we've got hydrogens and we got water being split over here, so lots of little things happening. So starting over here at the far left with our first photosystem 2, right? The first thing that has to happen is you have to have light energy striking photosystem 2. So light energy hits these electrons that are going to be excited within this photosystem, and that moves them to a higher energy state, and they naturally want to pass that energy along. And so that's what these arrows, these black arrows represent, that energy being passed along. Until eventually the chlorophyll, it gets past to this kind of central chlorophyll molecule right here. We call it this reaction center chlorophyll. It gets passed to this one, which elevates it to a point that it gets passed to a completely different carrier. Okay, so you have all of these little photopigments around, these antenna pigments around, and these are going to be things like the carotenoids, the chlorophylls, that are going to be absorbing light energy. And they're going to be kind of holding it on. They're going to pass it around, and then they're going to pass it to this central chlorophyll, which then can elevate it to that full status of being passed to a separate molecule. These electrons have to be replaced. These electrons that are getting passed have to be replaced. And so they split water. Water is split into hydrogens and oxygen, this half O2 molecule and this plus 2H plus. It gets split to those two different things, and the electrons holding them together are transferred to this reaction center chlorophyll, which can then, again, elevate it with that light energy and send it along to photosystem 1. Okay, so photosystem 2, all about splitting water. Okay, it's all about initially taking in this initial hit of light energy, kind of jumpstart this reaction. It's going to energize those electrons up. They're going to energize those electrons and then allow them to be passed along. But those electrons have to be replaced, and we do that with splitting water. Moving down the chain, these electrons get passed through this electron carrier. This is actually a hydrogen pump. Okay, so you can see that we have hydrogen molecules on the outside. We have hydrogen molecules on the inside from splitting that water. So it's taking a hydrogen, it's moving it up its concentration gradient, right? We're using energy, so this is an active transport. So we're using this energy from this high energy electron to increase the concentration of hydrogen ions within the thylakoid space, within this intermembrane space. And those hydrogen molecules are perfectly placed to then move down their gradient back out of the membrane through this ATP synthase pump. And so this one, it calls it synthetase. It's supposed to be ATP synthase like it is here. I'm sorry, I should have corrected. Wish I could have called that. But essentially, this first molecule is all about trying to make ATP, right? That we said that's one of the products we need to form for these light reactions, is that we need to form ATP. Well, here's how we're doing it. We're creating this hydrogen concentration gradient. We're creating a high concentration of hydrogen molecules, and then we're lining them up with this protein pump, this ATP synthase. And as hydrogen moves down its concentration gradient, it's going to be releasing kinetic energy, and that kinetic energy can then be harnessed into ATP. We're phosphorylating ADP, adding a phosphate to ADP to make ATP. We call it the chemiosmotic phosphorylation. It's a big fancy word, but let's break it down. Phosphorylation, adding a phosphate. Okay, that's not too bad. Chemiosmotic. This is referring to a chemical concentration gradient. We have a high concentration of hydrogen ions, and so they're moving down their chemical concentration gradient. So it's a chemiosmotic phosphorylation. Using that energy of movement of those hydrogen molecules to be able to add the phosphate to it. You ever see like hamsters running on a wheel, and then people use that to power a light bulb or something? It's the same kind of thing. We're using just the movement of these hydrogen molecules to spin this pump. Actually, if you see it in action, it actually rotates, and as it rotates, it adds this phosphate to this ADP. Again, just to use these terms, we're reducing ADP to ATP. Reduction is gaining electrons, reduction is gaining bonds. So we've reduced our ADP, and so we made that. Great. So now our electron, once it passes through this carrier molecule, it then goes towards our next photosystem, which is photosystem 1, the second one in the chain. I know. So by the time the electron gets here, though, it's really lost a lot of its energy. That transfer from through that pump, through that hydrogen pump, really took out a lot of its energy. So we get another hit. We get another hit of light energy. This excites these molecules. It passes it to another reaction center, chlorophyll. This one's a slightly different wavelength. This one's P700. The other one was P680. Those are the wavelengths that they're optimal at, right? And what we're doing here is that now we're, again, exciting this electron. Once it gets excited enough, it kind of hops off, right? Because that's what things with electrical charges do. Electrical charges will jump around. And so this electrified electron is going to jump over to this other molecule over here. And what do you know? Now here's where we form this NADPH. This is when we're reducing, reduction is gaining a bond, reducing NAD plus to NADPH. So it's gained this hydrogen bond with these two electrons. So that was the second thing we needed to make with our light-dependent reactions. We made our ATP in this step with the hydrogen pump and the ATP synthase enzyme. And then we just made NADPH by reducing this NADP molecule with those high energy electrons that we got from splitting this water molecule. So hopefully that I know it's a lot. There's a lot of things happening there. But again, go back to the big picture, rewatch this as many times as you need to, so that you can kind of understand, okay, light and water are coming in from this first photosystem. We're splitting the water molecule to harvest these electrons. We're exciting those electrons and then sending them through this hydrogen pump. This hydrogen pump then uses that energy of that electron to establish this big gradient of hydrogen ions that then move down their concentration gradient to make our ATP. There's our first product. That electron then moves to the next photosystem where it's re-energized, and then that electron gets used to reduce NAD plus to NADPH. Then we have our second product. And in the process, oxygen is produced as a waste from splitting water. So break this up into the two different processes as you need to. Come back to this one so that you understand, okay, we're making ATP, we're making NADPH, and then you can see how this gets used in the next reactions. And that's what makes these things a big cycle, because you can kind of see it down here, that the carbon reactions are going to use the ATP and NADPH. They're going to oxidize these back into their oxidized forms, NADP plus ADP, so that they can be sent back into the light reactions to get reduced again. And so it's this kind of, again, these are cycles. It's a cycle that's kind of being replicated over and over again, constantly inside of plant cells, almost constantly. We'll see. Okay, so once we have those molecules, we're ready to go into our carbon reactions. Again, carbon reactions are going to be pulling in carbon dioxide and fixing it into a sugar molecule. And we do this through a set of 3 steps in the carbon reactions. Yes, okay. So here is the carbon cycle. Again, I know when you first look at it, it's like, okay, there's a lot of things happening. There's a lot of stuff on this screen. But we're going to break it down into the three-ish, four kind of main steps of the carbon reactions. We call these different things. They have multiple names for them. The Calvin cycle is named after the person who discovered it. Carbon reactions are more common. Light independent reactions are common as well. That's another normal, a typical way of hearing them. What we try not to use are the dark reactions because technically the light dependent reactions require light. You require sunlight to excite those electrons. The light independent reactions, they don't really need light at this point. They need the products of the light dependent, but they themselves, the carbon reactions, can happen without sunlight. As long as they have ATP and NADPH, which of course require sunlight. So these are light independent, but the dark reactions, we'll see that some plants do this process at night as a way of conserving energy, but it's more accurate to call them light independent carbon reactions or the Calvin cycle if you want to. Sorry, just get a sip of water real quick. Okay, so again, we're going to break this up into these main steps of this cycle: carbon fixation, P-Gal synthesis, and I know there's one down here. We're going to kind of skip that for a second, and regeneration of RUBP. So 1, 2, 3 kind of main steps that we're looking at here that we can summarize this cycle into. First things to remember is that a cycle means that it's going to be reconstituting the materials that it needs to continue. So at the very end here, we're restarting with this RUBP molecule that we're going to talk about. So some molecules to highlight with this process are, of course, carbon dioxide. We have to have this carbon dioxide, this gaseous molecule, being brought in. So that is required. The stomata have to be open to be able to bring carbon dioxide in. This enzyme Rubisco enzyme, this is one of those, it's very, your textbook hypes it up a lot. It talks about how it's probably the most common protein in existence on Earth, because all organisms that use photosynthesis use this Rubisco enzyme, and they have to use a lot of it because, again, photosynthesis is not very effective, and we'll see why here in this process. And so the Rubisco enzyme is abundant in cells, and what it does is that it helps to trap that carbon. So Rubisco enzyme is going to synthesize, or catalyze rather, that process of fixing carbon to this, what we see this called this RUBP, Rubisco bisphosphate molecule. Okay, I'm sorry, I'm getting ahead of myself. I don't want to do that. So again, just kind of talk about the important things that you need to, the molecules that you want to know you're starting with: CO2, Rubisco enzyme, and RUBP. Again, Rubisco bisphosphate, and the enzyme is going to be combining these two things. So that's that first process. That first step is the carbon fixation. The Rubisco enzyme adds carbon dioxide onto a molecule of RUBP. And this creates an unstable 6-carbon molecule. And if your brain works like mine, I like to count the carbons. I like to kind of keep track of that as we're going along this process. So if you see this RUBP molecule, what we're starting with is 1, 2, 3. I'll click 1, 2, 3, 4, 5, and there's 3 of them, so that's 15 carbons. And then we have 3 more coming in: 15, 16, 17, 18. Okay, so now we have 18 carbons. If we look at this molecule, this unstable intermediate that Rubisco enzyme is kind of forming, it is 1, 2, 3, 4, 5, 6 times 3 is 18. So we're conserving our carbons, right? We have 18 carbons coming in, being combined, and we have this 18 carbon intermediates being formed. So again, these are unstable though. It does not like to exist in this 6-carbon form, and so it automatically almost splits up into this molecule, these PGA, right? So this next step is this P-Gal synthesis. I know you're like, we're already on step 2. Yes, we're already on step 2. And so in the P-Gal synthesis, what's happening is that this first intermediate, this first 6-carbon intermediate, is going to immediately split into 6 3-carbon intermediates. called PGA, phosphoglycerate. I'll say it one more time, phosphoglycerate. And so if you count, 6 times 3 is 18, 3 times 6 is 18. So our carbons are still right on where they need to be. But this PGA molecule, it's more stable. It's stable as it is, but it's not really useful for us. It's not what we need. We can't really do much with this. So we need to do pGal synthesis. pGal is phosphoglyceraldehyde. So PGA, phosphoglycerate, pGal, phosphoglyceraldehyde. And so to be able to make that, we have to use that ADP and NADPH. The energy from the ATP, we split off and we remove this phosphate, and we can use that splitting, that decomposition, catalysis, oxidation, whatever term you want to use, right, of that phosphate bond, and the electrons provided by the NADPH. We can use those two things to create this pGal, this PGAL. PGAL is our goal, honestly. I know we say that carbon fixation, we're trying to produce sugar, but sugar comes from this pGal molecule, this carbon molecule, right? Glucose, if you remember the formula for glucose, C6H12O6, it's a 6-carbon sugar. Well, pGal is a 3-carbon molecule. So what's going to happen is that this pGal molecule is going to split 5 of our 6 that we produced are going to go into the rest of the cycle, whereas one of them is going to split off. It will combine with another one. Now you have 2 3-carbon sugars coming together to make a 6-carbon sugar. That sugar, of course, can then go through the multiple pathways. Sucrose for energy, starch for storage, or cellulose for structure. Okay. Again, we're producing our 6 pGal molecules. Those are going to be splitting because one goes towards making a sugar. So it takes, just to kind of click into your brain, it takes 2 rounds, 2 turns of the Calvin cycle to produce 1 sugar molecule. Because you're only getting one pGal per turn that gets reserved towards sugar process. The other 5 have to go towards the regeneration of our UBP. Right? It's a cycle. This thing has to continue itself along. And so it has to regenerate its starting products. So the RUBP has to be regenerated, reformed, and we do that with 5 of those 6 pGal that we made. Again, it's a little bit more ATP from our light energy reactions, right? We use that, we oxidize that, send it back to those light-dependent reactions to get reformed, and then we use these 5 pGals, 1, 2, 3 times 5 is 15 carbons, 1, 2, 3, 4, 5 times 3 is 15 carbons. Right, so we use this ATP to kind of reshuffle these pGal molecules around, these carbon molecules around, and to make the RUBP again. There's a video on Blackboard. I believe I have this in the Chapter 5 resources. This TED Ed Calvin cycle one, it's really good. I wouldn't recommend watching it right away. You want to have a little bit more of an understanding about these processes a little bit more, so maybe watch some of these first videos. But this one I think is very interesting because it really explains this shuffling of carbons in a really clear way. So I really enjoyed that video. But for now, just kind of, again, thinking about it in terms of the main steps: carbon fixation, pGal synthesis, regeneration of RUBP. You have carbon dioxide being combined with RUBP by this Rubisco enzyme. And it makes these intermediates that then get passed along. Right? Oh, I guess I didn't click through, sorry. Right? We make our multiple pGals, 2 turns of the cycle gives you pGal molecules. Of course, right, sometimes, like this is showing it, this kind of makes it seem like these things are all happening within this nice little compartment. These molecules are bouncing all around each other. So maybe from this one turn, 2 of these 6 pGals actually did combine to become a sugar, and then you had 4 that got sent around, and then a 5th one from another turn of a cycle got combined too. It's not like it's as neat and tidy as it looks in here. Cellular processes are chaos, but there's order to the chaos. There's logic to it. This is just showing that regeneration of RUBP. to be able to start the cycle again. So we talked about the fate of glucose, right? It has multiple avenues for where it can be turned into. Sucrose for quick energy. So sucrose is just a disaccharide that can be split pretty quickly and used as glucose. Starch for energy storage, longer energy storage. Think of those starch products. And then cellulose for structures. Things like leaves, stems, roots, stamen, all those. Okay, so I'm just going to go back a couple of slides real quick. Hopefully, having this as a video form of me doing this means that you can go back, you can rewatch it as you need to. Because again, I know that these processes can be really overwhelming. There's a lot of terms that's being thrown at you. So as you need to, I'm just going to flip back a little bit. Go back to these kind of, where's the one I want? This one. Go back to this kind of big picture graph, and there's one that you can fill out on Blackboard. I believe it is this one. Yes. So this one is a labeling activity, so you can go in here and you can label the different structures of the thylakoid, the chloroplast with this process, kind of adding in what's going in, what's coming out. Hopefully that can help you organize some of this material. Because if you don't understand the big picture, the subtle nuances are really going to throw you. So start with these big pictures, get the overall reactions, and then start to go into the specifics of these processes and try to write them out and chart them out as best you can. Okay, so what we've been talking about is the normal Calvin cycle, carbon cycle, right? Whatever. It occurs under ideal conditions. So think about what most plants are going to like: more of a mild temperature, nice, lots of moisture. A few plants, are going to say no to moisture. Even cacti like a little bit of water, and they have a method of making sure they don't absorb too much, right? So for most plants, they go through this normal process of the Calvin cycle. When temperatures start to get too extreme, when they start to get really hot, when you start to lose any kind of water availability, or your water availability starts to go down, plants go into what's called photorespiration. And photorespiration is when plants actually close their stomata. In normal good weather conditions, their stomata are open so that carbon dioxide can come in, right? So it can be fixed as sugar, whereas oxygen can leave as its waste product. And so again, temperatures are ideal. Leaf cells are exposed to high carbon dioxide, low O2. Rubisco adds carbon dioxide. Cell maximizes sugar production. This is great. This is what we want. This is like when we are at our optimal level. You're at your maybe just resting or even light exercise, but you're still breathing easily and you're still doing just fine. Nothing's really going wrong. Photorespiration, again, the temperature's going up, water availability is going down, and so the stomata are closed to conserve water because plants don't want to get dehydrated, right? Plants, if water availability starts to get low, plants start to shrivel up, they start to wither. That then starts to lower their leaf surface area, so they can't get as much light, so they can't produce as much energy, and so it's a whole kind of cycle of processes. So during these more extreme conditions, stomata start to close, but that then limits how much carbon dioxide, it kind of stops carbon dioxide from being brought in and oxygen from leaving. Because the cells are still going to get a little bit of sunlight, and so they're still going to be exciting those electrons and splitting water molecules and producing oxygen. But what happens to that oxygen, right? It can't leave, it can't exit the cell. And unfortunately, Rubisco will bind to oxygen as well. It's, we in lab, we talked about the specificity of enzymes. And so the fact that Rubisco can bind CO2 and O2, just maybe think about, what does that tell you about the active site of Rubisco? What is the active site of Rubisco really binding to here if it will bind CO2 and O2 fairly equally? So think about what that says about the active site of Rubisco. But really, what happens during photorespiration? It's kind of like when we go into our lactic acid production, right? We lose our ability to bring in oxygen. You start kind of gasping for breath. Your muscles start to burn. Rather than kind of going through our normal energy pathways with oxygen, when we run out of oxygen, we start to go into that lactic acid production. And so plants do something kind of similar. They go into this photorespiration, where instead of bringing carbon dioxide in and fixing carbon dioxide, they fix oxygen. But unfortunately, that does not produce sugar. This is kind of an end pathway. Once you fix O2 to Rubisco, to the RUBP rather, RUBP and O2 just leave the Calvin cycle. They don't form anything, and then that RUBP can't be reformed, because remember, that's part of the Calvin cycle, is that RUBP has to be regenerated. So if you have oxygen binding RUBP and then that leaving the cycle completely, it can't then be regenerated, and so plants really start to suffer from that. They have to completely regenerate those molecules somehow. So plants have a very kind of strong incentive to be able to limit the photorespiration that occurs, right? So to do that, they've adapted multiple pathways. We call them carbon fixation pathways. What we've been looking at are the C3 plants. Most plants, 95% of plants on Earth, use the C3 pathway. It is by far, again, think about that. 95% of plants, all plants on Earth, use one same pathway. That's wild. Think about how big and varied life on Earth is. 5% is still a lot. 5% of plants on Earth is still a lot of plant mass, a lot of biomass. But the fact that 95% across all the different biomes of Earth use the same pathway is, again, I always just think about the evolutionary significance of those things. So again, we see normal C3 plant during the day. If sunlight is shining, they're getting light energy. They have good water availability. They're bringing in, their stomata open, they're bringing in CO2 into their mesophyll cells. There's a little plant anatomy. You might need to review your plant anatomy. They're bringing CO2 into their mesophyll cells. It's being fixed to Rubisco, to RUBP, and made into sugar. Okay, when it becomes night and there's no more light, this process stops. This all happens in one cell and one nice single pathway, very efficient. But not all plants live in conditions that are optimal for them. Some plants live in hot, dry weather, and so they have to adapt. So there's the C4 pathway and the CAM pathway. So C4 plants, things like corn, you can see this kind of these two green boxes is showing us that they've separated them into two separate cells. And so they still do everything during the day, right? The sun is shining. They maybe don't have quite as much water as they need to, but they're still doing okay. So they bring carbon dioxide in. They fix it one time into this molecule called oxaloacetate. You can abbreviate that OAA, OAA, oxaloacetate. They fixed it as that. It actually is going to get turned into something else called malate. We'll see. It's a more stable form of it. But they fix it once, they send it to this other cell, this bundle sheath cell, where they fix carbon again. So they fix carbon twice. There's two carbon fixations in this one. And this intermediate oxaloacetate is a 4 carbon, so that's where it gets the C4 name, is because it's a 4 carbon pathway, a 4 carbon intermediate. So knowing about C4 plants, they fix it twice. CAM plants, as you can see, it's happening at night. It's all in the mesophyll cell. We got one green box, so it's all in the mesophyll cell. You make your carbon, still 2 fixations, once as a 4 carbon molecule, and then actually it stays as that until daytime, and during the day, it's going to start producing sugar. It's going to fix carbon again during the day. So CAM plants are very unique. Let's see, I think we have a couple of slides with this. So going back to C3 plants. C3 plants do it all at once. During the day, their cells, right, within their mesophyll cells, this is where that carbon fixation is going to happen. But for C3 plants, it all happens in the mesophyll cells. So photosynthesis occurs in mesophyll cells. Look how loose they are. They're out here kind of towards the surface of the stoma. This allows them to get that direct access to carbon dioxide. And so they produce their sugars, and then their sugars can kind of diffuse along through these bundle sheath cells. And the bundle sheath cells surround a vein, and the vein is where glucose can actually be transported. Glucose, sugars in whatever form that it is. So C3 plants have their mesophyll cells, their bundle sheath cells, and their veins. C4 plants have the same thing. They have their mesophyll cells on the outside, but notice how they're much more compact in these kind of circular ring arrangements. And then deep to those are the bundle sheath cells that immediately surround this central vein. So somewhat similar arrangement with the vein and the bundle sheath cells, but in the C4 plant we have the mesophyll cells really packed tightly around the central structures. And so in the C4 plant, they're going to fix carbon once in the mesophyll cell, pass that into the bundle sheath cell where it gets fixed again, and then that gets transported into the vein. And again, C4 plants are doing this to try to reduce the oxygen exposure, because a lot of times their stoma are going to start to close and they don't want to let oxygen get built up. And so by fixing it once in the mesophyll cells, right, oxygen can build up in here. Oxygen doesn't get transported into the bundle sheath cells. Oxygen is the bundle sheath cells are kind of immune to oxygen, or impermeable is a better word. Immune. They're impermeable to oxygen, and so they don't have to worry about oxygen competing for RUBP. They can take the 4-carbon oxaloacetate that's produced in the mesophyll cells, and they can fix that in their Calvin cycle. Again, with our C3 plants, the stomas stay open because they have generally good conditions, and so as oxygen is produced, it can diffuse back out, and so they don't have to worry about having their mesophyll cells, their bundle sheath cells exposed to oxygen because it's leaving anyway. CAM plants, like I said, they do it a little differently. They divide the labor temporally. Sorry, I know I switched through that last slide, and some of y'all might freak out about that. This one was talking about the C4 plants. I talked about this on that previous slide when it was highlighting C3 plants. This is just kind of saying what I was talking about, how the light reactions occur in the mesophyll cells, the Calvin reactions occur in the bundle sheath cells, and so that isolates the Calvin cycle from being exposed to oxygen. So reducing photorespiration, that's what it's all about. So CAM plants, this is that other pathway that I keep saying we, like we're plants and we photosynthesize. We definitely do not. So only about 1% of plants are CAM plants. The reactions occur all in the mesophyll cells, so they don't have like the C4 plants where they separate mesophyll and bundle sheath cells. It's like C3 where everything is going to happen in the mesophyll cells, but it's also like CAM, or also like C4, in that there's this intermediate, this 4-carbon molecule fixation that happens before the carbon cycle itself. The way that they're different, that CAM plants are different from C3 and C4 plants, right, besides being kind of a blend of the two, is that CAM plants are going to fix carbon at night. So again, think about the desert. It gets much cooler at night, and so they can open their stoma. They can allow carbon dioxide in. Any oxygen that is accumulated, they can allow that to diffuse back out. And then during the day, they close their stoma, right? But at night, they've trapped all the carbon dioxide that they need to. So during the day, they close their stoma. That allows them to then start absorbing light, going and sending their carbon dioxide through the Calvin cycle, right? The struggle that CAM plants then experience is that once they've used up, if they don't, however much the carbon dioxide they trap at night, that's all that they have available to produce sugar for that day. So it could be that by 3 or 4 p.m., they've been exposed to enough light that they've already used up all of their sugar, and there's hours and hours of daylight that they're just missing out on, not being able to produce sugars. However, that makes up for, it's probably made up for in how much water they would lose if they kept their stoma open during the day. So it's a trade-off. It's a sacrifice. Again, none of these processes are fully efficient, and so the plants just kind of have to decide what's the least harmful, least energy loss pathway that we can do here. Okay, so that was pretty much it for the lecture for photosynthesis. We're going to review this stuff on Tuesday as well, but yeah, Tuesday. Expect a quiz. There's probably going to be a quiz on Tuesday. I'll find there definitely will be a quiz on Tuesday, what I'm saying. Definitely going to be a quiz on Tuesday. When it comes to the pathways, right, remember the overviews of them. Understand that there are different pathways and why they have different pathways. The other plants are adapting to this weather. I'm gonna fix this right here because that says C3 and it definitely is not C3 in this case. It is supposed to be C4, and it's not letting me edit. Weird. Huh. So don't read. Don't. Sorry, I just noticed that this said C3 and cam plants are adapted to hot dry weather, and it definitely should be saying C4 in cam plants. But I don't know how to fix that. It won't let me edit it for some reason. Again, that's why you always have to, I always say for your textbook stuff, for your worksheets, if you say, hey, no, this seems wrong, this seems conflicting, let me know, because it may be wrong. People write these textbooks. But like I was saying, make sure you understand the main differences between these different pathways, why they have these different pathways to adapt to different weather conditions so they can minimize photorespiration. Understand that C3 plants do everything in one cell, one fixation. C4 plants, 2 cells, 2 carbon fixations. And cam plants, they do it night and day with 2 carbon fixations in one cell. So just some lots of different pathways. Okay, thank you so much for your time and attention. Hope you have a lovely rest of your week.